Professor, my name is Halima and welcome to my oral presentation. Okay, here it is. What's my project name? The type of which I am interested is in FOXP2 gene. It is a gene that encodes a transcription factor, which is protein that regulates the expression for other genes. And the gene function is the CST complex protein subunits is encoded by this gene. The CST complex is involved in telomere maintenance and DNA replication. So here, what's the main goal of my project? So I am going to insert FOXP2 gene into a pcDNA 3.1 vector, which is designed for expressing genes in mammalian cells. The vector with the inserted FOXP2 gene is then introduced into HeLa cells, specifically HeLa cells with some modifications or mutations. Now we are going to the next slide. And what I'm going to show is to insert a gene into an expression vector. And for this particular approach, I am using horse-start PCR and the QIA quick gel extraction kit. The horse-start PCR is employed for specification gene application, minimizing non-specific products. The amplified gene is then dissolved on an aerosol gel, and the QIA quick gel extraction kit is used for efficient purification involving gel excision, binding to a specific membrane, washing, and elution. It will purify my gene of interest, and it is then ready for precise insertion into the expression vector. For this step, the registration enzymes are applied to create compatible ends on the both end of the gene and the vector. I will treat the resulting fragment with NEB buffer and ligate it using NEBT for DNA ligase. This combined approach ensures targeted DNA amplification, high-quality DNA purification, and controlled cloning into the expression vector. Here, the bacterial transformation is a crucial step in amplifying the expression vector carrying the inserted gene. For this, we purify the plasmid containing the gene of interest, and then it is introduced into the bacterial cells. The common use for this purpose is E. coli. Here's a step-by-step description of bacterial transformation and the subsequent selection of colonies containing the desired gene. Then the following day, I collect 5 to 6 bacterial colonies containing the transformed plasmid and apply the gene Z plasmid kit for plasmid DNA isolation. Then I purify the obtaining plasmid DNA using thermogene Z spin columns, subsequently perform extraction enzyme digestion using Hinton and ASCI on the isolated plasmid DNA. Anticipating a 300 BPV fragment from Hinton cleavage and a 4900 BPV fragment from ASCI digestion. Then I confirm the presence and correct size of the DNA fragments by analyzing them through aerosol gel electrophoresis. Additionally, incorporate a Sanger sequencing 12-step to validate the sequence of the inserted gene with the plasmid. Here in the final step, I will use systematic approach. This involves determining a kill curve for optimal selection agent concentration using lipofructamine 2000 for enhanced transfection efficiency. To visualize the expression and localization of the FOXP2 protein, anti-plaque antibodies like DYK tags are applied. Applying techniques like immunofluorescence or Western blotting. Finally, telomeric measurements are conducted using a Q-FISH technique, providing a quantitative analysis of telomere length and offering insight into the impact of the expressed gene on telomeric dynamics in HeLa cells. The comprehensive strategy ensures precise transfection, effective selection, visualization of the expressed protein, and detailed study of potential effects on telomere in HeLa cells. So thank you so much, Professor. I hope, thank you so much for listening. Thank you.